mode.
Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled RNA Biomarker Development, From Sample to Insight. My name is Michelle Tran, and I'll be your X-Talks host for today. Today's webinar will run for approximately 90 minutes and includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation. Use the questions chat box and we'll try to attend to your questions during that Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank QIAGEN, who helped develop the contents of this presentation. QIAGEN is committed to enabling its 500,000 customers around the world to quickly and reliably reach their goals of useful, actionable insights. The company offers innovative solutions that cover every single step along this journey, not just technologies, but bridges from samples to insights. Sample to insight means QIAGEN offers the industry's most reliable sample technologies. Top quality assays and panels that enable accurate analysis and identification of diseases and genetic variations. Bioinformatics software and curated knowledge bases which transform raw data into relevant, actionable findings, as well as automation solutions which provide seamless and cost-effective workflows. The insights gained may lead to one small step or a giant leap forward for science and healthcare. And now I'd like to introduce our featured speakers for today's event, the first being Dr. Eric Lader, Senior Director of Product Development at QIAGEN. Dr. Lader is a molecular geneticist by training and is currently Senior Director of Product Development at QIAGEN. In his 13 years at QIAGEN, Dr. Lader has led development of tools for functional genomics, gene expression profiling, microRNA profiling and characterization, as well as gene read targeted panels for next-generation sequencing of DNA and RNA. As the inventor of RNA Lader, Dr. Lader is a pioneer in the field of nucleic acid stabilization for basic research and molecular diagnostics. Our second speaker for today is Dr. James Deere, Senior Clinical Lecturer in Clinical Pharmacology, Royal Infirmary of Edinburgh, Edinburgh University. In addition to his current role, Dr. Deere is Chief Investigator on the Markers and Paracetamol Poisoning Study, a UK CRN portfolio study funded by MRC. Dr. Deere is also a co-investigator on the Scottish and Newcastle anti-emetic pretreatment for paracetamol poisoning study. And now, without any further ado, I'd like to pass the presentation off to Dr. Eric Lader. Dr. Lader, you may begin your presentation when ready.
Thank you for that kind introduction. Let me make sure I'm showing the right thing here. Yeah, okay. What we're going to do today is I'm going to give you a sort of a broad overview on using RNA tools and technologies as a basis for biomarker development, and then hand the talk over to Dr. Deere, who will actually show an application of using this to benefit people in terms of developing a molecular diagnostic. Just for bookkeeping purposes, this is my email address in case anybody is interested in contacting me after the webinar. And as a final bookkeeping note, I'm required to mention that the products described in this webinar are intended for molecular biology applications only, not intended for the diagnosis, prevention, or treatment of a disease. So I'd first like to start with the basic question of what is a biomarker. A biomarker is a characteristic that is subjectively measured and evaluated as an indicator of either a normal biological process, a pathogenic process, or a pharmacological response to a drug. And so it's essentially a proxy for something that's going on inside of your body that can be objectively measured. And some of these can be presence of antibodies, which can be detected and measured by an ELISA assay, an abnormal blood pressure, abnormal blood cells, abnormal electrolyte levels, histological indicators which can be determined by microscopy, abnormal liver or kidney functional markers, so enzymes that might be at aberrant levels in the blood, presence of a muscle injury, protein marker that might indicate damage to heart muscle, or what we'll be concentrating on today is gene status, change in gene status or change in gene expression status. And the methodologies that are used to quantify these changes are typically real-time PCR-based, next-gen sequencing-based, or hybridization array-based. So turning specifically towards nucleic acid biomarkers, I view these personally as the gateway to personalized medicine. And the reasoning behind that is that nucleic acid is essentially a stable universal analyte. Sequences are different. The chemistry of DNA and RNA is slightly different, and we'll go into some of that later. But in general, nucleic acid is nucleic acid, which means we can apply universal detection technologies rather than, for example, developing different antibodies and different ELISAs for different molecules. We have a sort of a stable universal platform by which we can measure these analytes. Also, in regards to nucleic acid, we can achieve single molecule sensitivity and extreme specificity, so we can tell when there's a single base change in a sequence, for example, that causes us a somatic mutation and can lead to oncogenesis. These biomarkers, just as an example here on this slide, what you see are a list of several of them that can fall into the categories of predictive, diagnostic, prognostic, and theranostic biomarkers. So in terms of prediction, one can assess whether there's a germline inherited mutation in BRCA gene which would predispose someone to breast cancer. Diagnostic, we can look for a BCR-ABL somatic translocation either at the DNA level, or we can look at the RNA level for a fusion transcript. Prognostic, when we sample a breast tumor by biopsy, we can look for a HER2 somatic amplification, or we can look by immunocytochemistry for overexpression of the HER2 protein. And finally, for theranostics, we can look at germline SNPs in CYP26 genes, which would influence how your cells would metabolize a particular drug, or, for example, a chemotherapy reagent in terms of how you would respond to that drug. One more term I would like to throw out there is a non-invasive biomarker. So this is where all the excitement obviously is in diagnostics, and that is to develop biomarkers that can be used as routine diagnostics, and these should be non-invasive. So on a chart of more invasive to less invasive, what you see here is an invasive biomarker might be a surgical biopsy. These are invasive, but they're very prevalent. Obviously, when there's solid tumors, typically fine needle aspirates are taken for analysis. A little bit less invasive would be cerebrospinal fluid. Blood samples are obviously a standard in the industry for sampling of a patient, and while it's slightly invasive, it's typically met with high acceptance by the patient community. A drop of blood, for example, to measure sugar levels in a diabetic, and ultimately the least invasive biomarker would be a cheek swab for DNA, or sampling of urine or saliva or any other body fluid that's easily accessible to look for biomarkers. In regards to blood, which is a major a source of material to develop biomarkers, clearly there's a cellular component which would contain what's normally found in blood in terms of the PBMCs, endothelial cells that are shed, along with red blood cells, other white blood cells, but also there can be circulating stem cells or circulating tumor cells, which are generally at very low levels, which can be affinity purified and then evaluated. In the serum and plasma, which we'll talk more about, there's a whole host of nucleic acids that can be found that are either shed by cells that line the circulation or are added to the circulation by, for example, tumors which have a very well vascularized structure, also ECM components and lipids. So a biomarker can be one or a combination of different materials that can be found in blood and then developed into a test that can predict a disease. If we do a PubMed search, essentially for gene expression and biomarker, or RNA and biomarker, what you find is an explosive growth over the last 10 years, essentially a logarithmic or exponential growth of publications. And to date, we're over 22,000 peer-reviewed publications which have in their title or abstract essentially RNA or gene expression along with biomarker. And a lot of this growth has been led by the adoption of high-throughput technologies such as NGS and high-throughput real-time PCR, and also led by the discovery of stable nucleic acids in biofluids and in whole transcriptome profiling of gene expression in solid tumors. So in regards to RNA in body fluids, and I think James will also show a slide very similar to this later on in his talk, it was quite a surprise a couple of years ago when we found that there were nucleic acids that were present in the extracellular compartment in blood. Considering the cellular compartment, obviously there's RNA and DNA in neutrophils and eosinophils and other white blood cell components. There's some residual nucleic acids in platelets, which are protected from any nucleases in the serum or plasma by their membranes. There's clearly a lot of erythrocytes. We all know that there have been circulating tumor cells that have been found in the cellular compartment. These are at pretty low levels, but they do contain novel the protein and RNA and nucleic acid content of the tumor that they were sourced from. But that discovery of nucleic acids in the acellular compartment was fairly surprising. If you take a naked nucleic acid, a naked RNA, and you put it in a serum sample, it will degrade in minutes, in moments. Maybe it shouldn't be all that surprising that there's really not in nature such a thing as a naked RNA. RNA is typically both folded up into a secondary structure, but also complex with proteins when it's found inside of a cell. And presumably when cells lyse or otherwise their membranes are disrupted, RNA can be spilled into circulation, and these would be complex with proteins. But there's also this component of exosomes and microvesicles, and these small particles have been known and characterized for many, many years in terms of their structure and their origin as budding off of cells and their containing protein from the surface of a cell that they're shed from. But the amount of and the variety of RNA and DNA that's carried within them was fairly big surprise last couple of years. And again, these are microvesicles or nanovesicles, or smaller than microvesicles. They're budded off cell membranes, so they carry with them on their surfaces proteins that were found on the surface of the progenitor cell. Within them, it's not entirely surprising that you'd find RNA that was also in the progenitor cell. People are also finding DNA, which is a little bit strange in terms of where it comes from. It's generally fairly small. These could be coming from cells that undergo apoptosis. It could be that when the nuclear membrane is disrupted in replication, some DNA somehow ends up in a microvesicle. It's not really clear. But nonetheless, there's nucleic acid in these. They have a lipid bilayer. On that bilayer are proteins that are specific to the parent cell. So you could imagine doing immunopurification of these and identifying and collecting them based on these surface proteins and analyzing the material within them to get an idea of what was going on in that progenitor cell, especially if that progenitor cell was a cancer cell. So these are good for us in terms of delivering nucleic acids from one site in the body into the circulation where we can detect and quantify them. They also have what appears to be a very important biological function in normal cells, maybe the best example of which is that the placenta sheds huge numbers of exosomes into the maternal circulation, which appear to affect the host versus graft, or the host versus placenta reaction during pregnancy and suppress that reaction. So there's a normal function of exosomes in circulation. But exosomes also seem to be able to function in other ways as remote intercellular mediators, or as what they're commonly called as proxysomes. So they're proxies for cell-to-cell interactions, which is extraordinarily interesting, especially since it's found that many cancer cells shed large numbers of exosomes. And these have been shown to have function both in vitro and in vivo. And so, for example, if you take a very metastatic and aggressive cancer cell line, grow them in tissue culture, isolate the shed exosomes from the growth media, and inject those into an animal which is containing a tumor which is normally non-invasive and non-aggressive, the presence of these exosomes, which carry along with it nucleic acids and proteins from the aggressive cancer that was grown in cell culture, can confer greater likelihood of metastasis of the cancer that was growing in vivo in the animal. So it certainly does appear that exosomes shed from cancer cells have a role in metastasis and growth and colonization of those cells elsewhere in the body. So I'd like to talk a little bit now about RNA isolation technologies, where we approach the RNA content from both cells and from body fluids. For the basis of this discussion, we're going to condense the QIAGEN RNA isolation portfolio down to just a couple of products that represent the whole family. And one is microRNA-seq, which we use for isolating total RNA, including small RNAs like microRNAs, from either body fluids or cells or tissues. I won't be speaking specifically about it, but I want to mention the QIAMP circulating nucleic acid kit. This is designed specifically for isolating nucleic acids from circulation, both DNA and RNA. And the difference between it and microRNA-seq is that it can tolerate much larger sample input volumes. So if you're looking for a very rare transcript and you need to sample a much larger amount of serum or plasma or urine, QIAMP would be the way to do it. And I want to briefly touch on exoRNA-seq, which is a modification of the microRNA-seq family designed for exosome purification and for isolating the RNA content of these purified exosomes. So as a description of the RNA-seq and microRNA-seq family, I'm going to turn to looking at the microRNA-seq serum plasma kit. This is called this for a couple of reasons. The kit includes a synthetic RNA control for normalization. So in all of your samples where you're going to be isolating nucleic acid, you can spike in a synthetic RNA to monitor sample-to-sample variability in isolation, but it also allows you to assay that spike in by PCR and look for inhibitors of the RT-PCR chemistry. So blood often, or blood products often, can be collected in heparin. That, for example, is a very potent inhibitor of reverse transcriptase. It's good to know whether your samples are contaminated or not. And so this internal control will serve to monitor that. The kit is also designed for rather small volumes. You can isolate nucleic acids from a microliter of serum or plasma, which actually is enough to do an experiment, up to around 400 microliters, which is enough to do a very large experiment for microRNA. If we're looking at some very rare mRNA targets, we may have to boost that volume up or do a preamplification, which I'll mention on an upcoming slide. Our elution volumes are optimized for molecular biology applications, so we're eluting in a relatively small volume, and that whole volume can be used, for example, in a reverse transcription reaction. It's easy to do, and the procedure is automatable on either a QIAcube or other robot. And the way the procedure works is we do an organic extraction in Chiazol. We take the aqueous phase, and we do a bind, wash, and elute off of a solid phase, essentially a filter in a capsule. This can be done either in a tube or in a 96-well format, so they can be done in relatively high throughput. So as I mentioned, exoRNA-seq is a protocol, a sample isolation protocol to purify, selectively purify the RNA from exosomes and other microparticles from serum or plasma. So this part that's in the shaded box is essentially microRNA. And upstream of that, what we're doing is we're taking serum or plasma, we're doing a filter to remove large particulates. We're taking that filtered plasma or serum, and we're collecting the exosomes and microsomes on a special filter in a buffer that allows them to be captured by that filter. Then we're doing a wash and an elution. And the elution is done actually with chiazole, so we're lysing the exosomes and releasing the nucleic acids from the collected sample. There's been a lot of work done on this by our partner, Exosome Diagnostics, who have proven that this material is very, very much in line with the purified nucleic acid that you get from the canonical or gold standard method for purifying exosomes, which is ultracentrifugation. What else can I say about this? The microvesicle isolation is about 20 minutes long. The RNA isolation for microRNAs is about 35 minutes. And why you would want to do this is a question that I'm asked frequently. So when you look at the population of nucleic acid in serum or plasma or other body fluids, there's actually very, very little mRNA that's not in exosomes. If you're looking in particular at mRNAs in circulation, they're probably going to be in exosomes. And in order to get enough material to actually quantify them accurately, you typically, and to decrease sort of the background noise level, you would typically choose to do an exosome isolation. For microRNAs, the story is a little bit different. There's actually quite a bit of microRNAs both outside of exosomes and inside of exosomes. Their makeup is a little bit different. Stuff that's in exosomes typically is shed by cells and shed in great numbers by cancer cells. Material that's outside of the exosomes are often released during apoptosis. They could be released in a disease such as liver disease or kidney disease, or in tumors where there's a lot of apoptosis going on. So the choice of doing total RNA and looking at the microRNA component or doing an exosome purification generally would depend upon what microRNAs you're interested in, where they're mostly located, and whether one of the other procedures optimizes the signal-to-noise for your experiment. So what we generally recommend people to do is to do the easy thing first, which is to look at the total microRNA component, and if that doesn't work, if the background level of a particular microRNA is too high and is making it difficult to see the data that you're looking for, you could go ahead and try to do exosome isolation. So if we look back 10 or 15 years and you ask the question, or you looked in PubMed as to what RNA species were being developed as biomarkers, what you would see is almost exclusively mRNA. And these were found by, 15 years ago, not as much by whole transcriptome sequencing, but by qPCR analysis and by hybridization array analysis. Fast forward to 2015, and what we're looking at now is not just mRNA, so the 30,000 or so mRNAs that are found in humans, but the few thousand or so microRNAs and the tens of thousands of so long non-coding RNAs, which have been mostly found by next-gen sequencing over the last couple of years. So if you look at the human transcriptome versus the human genome, about 90% of the genome is estimated to be transcribed, but only 2% of it is translated as protein. So the majority of the RNA that's transcribed from the genome is not mRNA, but non-coding RNA. And this is the big difference between lower organisms and higher organisms. The higher you go in evolution, the greater the percentage of the molecules are non-translated RNAs. And we've been naive for many years in assuming that most of this stuff was junk. And in fact, it's clear now that most of this stuff is not junk, and that the expression of these RNAs can be regulated temporally and spatially specifically, and the dysregulation can be involved in disease. And that the total story here is not one of DNA to RNA to protein, but that the total RNA story is one of a very complex interaction of both coding RNAs and non-coding RNAs, small RNAs and long non-coding RNAs with each other, with proteins, and with physically with the genome itself. So long non-coding RNAs, by definition, are non-protein coding, and they're longer than 200 nucleotides. That's somewhat of an arbitrary cutoff. Things that are smaller than 200 are small non-coding RNAs. These are called, abbreviated as LNC RNAs or link RNAs, LINC RNAs, depending on whether they're between genes or within other protein coding genes. There are more than 20,000 different long non-coding RNAs, so their diversity is as large or larger than the entire mRNA transcriptome. Their organization is complex. They are either intergenic, so they're not within a protein coding locus, or they can be intragenic, contained within a protein coding locus. They can be sense or antisense to the traditional coding transcript in that locus. They can partially overlap with it. They can partially overlap backwards with it. If they're contained within the locus in the same orientation, they can be controlled by cis-acting regulatory elements within that gene. If they're elsewhere in the genome, their transcription can not necessarily be controlled by Pol2, but can be controlled by other DNA polymerases as well. Again, by definition, they don't contain an open reading frame, or at least they don't contain a standard open reading frame that we can identify. They may or may not contain a poly-A RNA. In experiments that we've done looking at profiling long non-coding RNAs before and after oligo-DT selection certainly shows that some of them don't contain poly-A and some of them do. Their expression is generally lower than mRNA taken as a group, so sensitive methods such as qRT-PCR are required for detection and quantification. And the roles of long non-coding RNA in biology are feverishly being studied and just being elucidated now, and so daily there's new developments. Everything you can imagine that a long non-coding RNA could do in a cell, essentially it will be found at some point that they're doing. So they can interact with the transcriptional machinery at an mRNA encoding locus and negatively affect expression. They can positively affect expression. They can hybridize to a pre-mRNA and result in changing the splicing pattern for that mRNA from its precursor. The antisense transcript, long non-coding RNA transcript, can hybridize to a sense strand coding transcript, make a double-strand RNA, and allow DICER to generate endogenous siRNAs. They can have multiple binding sites for microRNAs and act as microRNA sponges to soak up microRNAs and affect the translation of other genes. They can modulate the activity of proteins by binding that specific protein. They can alter protein localization in the cell. They can directly affect epigenetic processes and change imprinting patterns in cells. All of these things can obviously lead to pathological changes when things don't go right in terms of cancer or cancer progression. And obviously, the proper activity of long non-coding RNAs like mRNAs and microRNAs are essential for normal differentiation and development. So new and very exciting. MicroRNAs, the other non-coding RNAs that we'll talk about, are generally considered to be master regulators of gene expression. They're short, typically 21 nucleotide RNAs. They're conserved in all eukaryotes. They're expressed in all mammalian tissues. And changes in microRNA expression is correlated with pretty much anything you can imagine. Correlated with gene expression changes in development, differentiation, signal transduction, infection, aging, and disease. So they are just as intertwined with cellular processes as mRNAs are. MicroRNAs are transcribed by Pol2. They're either, again, intragenic or intergenic, but they do use Pol2 promoters so that they're regulated in as complex a manner as mRNAs are. Because they're transcribed by Pol2, they're transcribed as long primary transcripts. They're processed in the nucleus the way an mRNA precursor are processed, but there's a particular structure that's recognized by an enzyme called DRASHA that makes this hairpin pre-microRNA, which is then exported in the cytoplasm by the transmembrane protein exportin 5. This pre-microRNA with this hairpin loop on it is again processed into a mature microRNA, which is a short double-strand RNA. it's assembled into what's called a loaded RISC complex, or an RNA-induced silencing complex, in which one strand of this mature microRNA is actually assembled in RISC. The other strand is discarded. This is done by an ATP-dependent helicase. And then using the sequence of this one strand of this mature microRNA, the RISC complex is guided to typically the 3' end of an mRNA, where it will bind and inhibit translation, or it can also bind and cause degradation, or site-specific cleavage and then degradation. This process is conserved in all plants and animals, in eukaryotes. So it's very essential to gene regulation. And there are, at last count, conservatively somewhere between 1,000 and 3,000 different microRNAs that have been described in humans. The higher end includes a lot of sequences that have been found by next-gen sequencing and have not necessarily been proven to be specifically involved in gene regulation. So now that we've looked at RNA isolation and we've looked at the types of molecules we'll be speaking about, I want to just briefly touch on the tools that we use for real-time RT-PCR-based gene expression profiling, and in a very condensed way try to explain this in a way that will also be understandable. There's two different product lines that we have that address these three different RNA types that we've talked about. One is called the RT-squared profiler, which is used for quantification and screening of mRNA and long non-coding RNA expression. The other is called the microscript system, or the microscript PCR system. In particular, we're looking at microRNAs. Why these are different is generally because of the enzymatic steps that we need to do to quantify these molecules. MicroRNAs being extremely short require some chemistry modifications, essentially, to get it to work. So what are these arrays? What they are content. So QIAGEN has invested a lot of time and effort into the literature, essentially, and have chosen content to put on arrays that fit on any real-time PCR instrument that are biologically relevant. So we're not simply, for example, collecting kinase genes and putting kinase assays on an array. As you'll see in the next slide, we're looking at essentially text mining and database mining and putting together panels that are useful for researchers in terms of biology or disease. So you can screen a very relevant collection of genes rather than screening one gene at a time or having to do your own data mining and figure out what things are useful for your experiments. So these are technically relevant. And for mRNA, one thing that people may not be thinking about is that a lot of genes are regulated at the level of post-translational regulation, for example, phosphorylation. Those wouldn't particularly pay to look at the mRNA level, so we are only generally looking at genes that are regulated at the level of transcription. These are curated in-house and also curated in collaboration with thought leaders. Everything we do is covered in human, mouse, and rat, but we also cover a lot of other species a lot of other mammalian species, insects, as well as plants. And as I mentioned, these are formatted for your typical 96-well and 384-well PCR instruments, as well as the QIAGEN Rotogene and the Fluidigm Biomark for high-throughput gene expression analysis. So just some examples of content. We do have arrays that are focused on pathways. For example, apoptosis is very popular. Toxicology and drug, ADME is very popular, signal transduction. We have arrays that are focused on different aspects of neurobiology, but also a whole panel of arrays that are specific for different cancers and other diseases. On the microRNA side, the micronome itself is approachable in terms of high throughput screening, so you can actually screen the whole human, mouse, or rat, or other species of micronome. There's also more focused micronomes, for example, all of the microRNAs that are detectable in serum and plasma in normal samples, all of the microRNAs that are detectable in normal liver. You can screen all at once in a 384-well format. There are focused arrays. The numbers are smaller because less is known about microRNA function, but there are cancer-specific arrays and then pathway-specific arrays. When we go to long non-coding RNAs, while we have the entire long non-coding RNA genome of human, mouse, and rat available, there's much less known about their functions, so there's a lot less focus there. But we do have a few arrays, for example, a cancer-focused panel. So the way the system works, if you look down here, you can see a 96-well array. From well 81 to 84 are specific assays for specific transcripts. We have typically on an array housekeeping sequences added for normalization. On an mRNA array, these would be mRNAs. On a microRNA array, these would be short non-coding RNAs. There are controls for genomic DNA contamination and for the enzymatic reactions that are used in the procedure. We do an RT reaction. There's an optional preamplification for when our samples are extremely small. We essentially make a master mix, load the array, and run it and analyze the data on the QIAGEN website. So all of the data analysis is done on our website. You're essentially doing an RT reaction, loading, running, and doing an analysis. So same day experiment for any mRNAs, long non-coding RNAs, or microRNAs from any sample on any real-time PCR instrument. As an example, because Dr. Deere will be speaking about profiling from serum, this is profiling microRNAs in 3 different body fluids. So serum, urine, and cerebrospinal fluid. And in all these arrays, we're normalizing expression to the mean of expressed sequences. I don't have a lot of time today to talk about normalization. We certainly can discuss that afterwards. What you see on the panel on the left is unamplified, unpreamplified serum. So there's a lot of microRNA in serum, so much so that we can do complete experiments on microliter amounts of serum. So this is actually the average of a couple of normal samples versus a couple of patients with non-small cell lung cancer. And what you can see is that most of the expression of these microRNAs on this cancer finder array are expressed fairly similar, the average across these different people. There are some that are overexpressed. And the ones that are mostly overexpressed here, in fact, have been previously reported in the literature as being overexpressed in the solid tumor. So even in this very, very basic experiment, you can see that you can find microRNAs that are in the literature known to be overexpressed in the tumor that are in circulation and at aberrantly high levels. These other 2 are here just so that I can illustrate the preamplification. So typically when people are looking at nucleic acids from urine, they're doing massive amounts of material to isolate very small amounts of nucleic acid. We have a universal preamplification system that alleviates the need to do that. So we can do profiling on very, very small amounts of urine, which also opens up the door to do this on, for example, mice and rats. These are just 2 normal donors using a MyFinder panel. And what you see is remarkably, actually, that the microRNAs that are detectable in these 2 people are essentially the same. There's very little difference. There's some noise here. These are different people. They're not age-matched or sex-matched. There's 1 or 2 microRNAs that stand out as being, I guess, differentially expressed between them. But the majority of them are present in both of them and present at very similar levels. So what this suggests is that these are microRNAs that are in the non-cellular fraction of urine that are shed under normal circumstances by the cells that line the urogenital tract. And so that's pretty an acceptable explanation. You could imagine that if one of these people had a prostate tumor or one of these people had a bladder cancer, that you would certainly see a change in expression profiling of microRNAs in the urine. And in this panel, what we're looking at are CSF samples from 2 people. And again, remarkably, most of the microRNAs that are present in these samples are at very, very similar levels between these 2 non-age-matched or sex-matched people. They're basically 2 anonymous donors. There are a couple that are differentially expressed, but these are normal people. What this also means is that normally in CSF there are microRNAs that are shed constantly into CSF by the lining of the central nervous system. And you would imagine if there was brain trauma or brain injury that you might see differentially present microRNAs. So in the few minutes that I have remaining, I just want to go over an example of using sort of a combined approach to look at RNA expression as a biomarker. And this example is done by a couple of people in my lab looking at thyroid cancer. And so thyroid nodules are very common in the U.S. population. There's over 500,000 fine needle biopsies that are done every year, and these are examined essentially histologically to decide whether that nodule is cancer or if it's benign. There's been some attempt at developing a commercial classifier assay, but it's really sort of in the development phase. So we thought it would be interesting to look at this material and ask the question of whether it's doable to look at mRNA, long non-coding RNA, or microRNA in these samples to see if we can distinguish benign nodules from thyroid cancer. So we've actually looked at, this is frozen tissue from frozen sections. We've got a panel of benign hyperplasia or follicular adenoma samples, and then a panel of thyroid cancer, papillary thyroid cancer, and follicular variant papillary thyroid carcinoma, so normal and cancerous. Isolated RNA using the microRNA mini kit. For mRNA and long non-coding RNA, we use the RT² system as I described. For mRNA, we're actually using an array that was developed specifically for thyroid cancer. So this is an array that has targets on it that from the literature have been reported to be involved in thyroid cancer. For long non-coding RNA, we're essentially using the best of panel. So it's a panel that has the most well-known and studied and broadly described long non-coding RNAs on it. For microRNA, using the microscript system, we're using the MyFinder 384. So this is a panel of sort of, again, the greatest hits of microRNA that have the most information about them published in the literature. For data analysis, we're using the GeneGlobe Data Analysis Center at QIAGEN's website. And I want to just touch a little bit upon what you do next after that using QIAGEN's Ingenuity pathway analysis. So here's an example of mRNA profiling, and we're looking at the average signal of benign thyroid nodules versus thyroid cancer. Not too surprising that there are a lot of transcripts on this array that come up either upregulated or downregulated in cancer, because this array was in fact developed for genes involved in thyroid cancer. So there are 68 that are statistically upregulated significantly and 46 that are downregulated. And it's not surprising that a couple of these have been described as being involved in cancer or being described as being downregulated in cancer. That's what this array was developed for. When we look at the same samples for microRNA profiling on this 384 panel, what we find is a number of microRNAs that are upregulated, a number of microRNAs that are downregulated. And that if you look at the ones that are upregulated, there's some information in the literature about them being upregulated in thyroid cancer. So we've recapitulated that information. In MIR31, 5P and 3P designate the two strands of the same mature microRNA, which are upregulated about 100-fold in these samples versus in the benign samples. That's a major upregulation, and it does suggest that it's at the level of transcription because both strands of this mature microRNA are upregulated. A couple of microRNAs downregulated. These could be potentially tumor suppressor microRNAs, one of which, MIR211-3P, has already been reported to be downregulated and suggested to be a tumor suppressor in breast cancer. Turning to long non-coding RNAs, again, same RNA samples. This is a relatively small panel. Again, we know relatively very little about long non-coding RNAs and their function. There are 8 that are significantly upregulated. One is already reported to be an oncogenic long non-coding RNA. MIR31HD is over 100-fold upregulated in these samples. There are several that are downregulated. Link 00261 is known to be a tumor suppressor. PTCSC1 is already implicated in thyroid cancer as a tumor suppressor, so not surprising it's downregulated. And there are a couple of others that are that have been reported to be downregulated in other cancers and could possibly be acting as tumor suppressors. So all in all, a very interesting group of findings from a relatively simple experiment on a dozen or so samples. And now, yes, these are on tissue samples rather than on serum and plasma, but Dr. Deere is going to talk much more about that after me. If we take a random forest classifier model, we can actually, simply looking at these long non-coding RNAs that are differentially expressed, fairly well classify the benign versus the oncogenic samples based on just these long non-coding RNAs. We've got a positive predicted value of 0.9 with this small panel. And you can see we're fairly well separating the benign from the cancer. And this leads us to the question of why aren't people looking at combinations of RNA analytes, of mRNAs, long non-coding RNAs, and microRNAs to develop more sophisticated panels for biomarkers? And so can you integrate mRNA, microRNA, and non-coding RNA data to improve that signature? So imagine if we took all three sets of differentially expressed targets. If you do a literature search on this, what you find is that there are a couple of papers that are reporting trying to do this, essentially with microRNA and messenger RNA, and I've bolded that in all of these papers. So mRNA and microRNA, mRNA and microRNA over and over again, all in the last year or so, and essentially none that have tried to integrate also these 25,000 long non-coding RNAs into this. But obviously this is an area of intense interest, and people are clearly working on trying to develop a more comprehensive signature as a biomarker. And my last couple of slides, I just want to mention Ingenuity pathway analysis, which allows you to ask the question, what's next? So now that we know about these differentially expressed microRNAs, mRNAs, and long non-coding RNAs, we can use IPA to model and analyze and understand complex data sets. It lets you integrate gene expression, microRNA, and genomics, so somatic mutations, germline mutations all together. The IPA database actually is hand curated by PhDs, and there are 5 million relationships that are curated and reported in this relational database. It allows you to look at upstream regulators and downstream targets of your differentially expressed targets, look with search engines that can find molecular interactions and effects on cellular phenotypes. And all of this information, by the way, there's a really nice video on the Ingenuity website that is far better than I am at explaining this, and it's at ingenuity.com/product/IPA. So in this example, what we're using is what's called the microRNA target filter. So we're taking the microRNAs that are differentially expressed in these samples and employing a couple of filters and then a search. We're looking for mRNAs that are have an inverse expression relationship, but are differentially expressed. So which mRNAs go up when the microRNAs go down, and which ones have microRNA binding sites in their 3' UTRs. And then also looking for ones that are involved in cancer and or endocrine disorders, and ones that are known to be expressed in thyroid and some select signaling pathways. And you can see for some of these microRNAs that are up or down regulated, you can see that these are upregulated. They're experimentally observed to be upregulated. There are mRNA targets that are experimentally shown to be inversely regulated. They are involved in cancer, and they are expressed in the thyroid gland. So you can start building pathway maps showing how these microRNAs and mRNAs can interact together in cancer. And so if you try to do that, what you see when you map these mRNAs and microRNAs together, that what we get is a predicted activation of a physiological response of the invasiveness of cells, which is what you expect in cancer versus normal, right? That they would be more tending to be invasive and metastatic. If we add the long non-coding RNA differential expression into this mix, it's a much more complicated map that the software draws, but you also see that a primary predicted effect using this molecular activity predictor is on increasing metastasis of these cells. So we've done an experiment here using 3 commercially available content arrays, a number of samples that were commercially purchased from sample supply for benign or oncogenic or cancerous thyroid cells, and developed quite a model. So this is, to me, is very impressive, and it should be very impressive to you as well. So my summary is that we've demonstrated that there is a complex interaction of coding and non-coding RNAs, so we can't simply look at one species of RNA anymore, that we can enable you to engage in total RNA discovery, and between the assays and the reagents and the Ingenuity IPA, you can get a deeper understanding of the functional relationship of these molecules to each other. And so essentially any sample, all samples, any RNA species, all RNA species, all of the tools to do the primary data analysis and the interpretive data analysis available online at QIAGEN. And hopefully that you can use these tools to turn hypotheses into actionable insights. And just a final word before I turn this over to James, and that is that any gene-specific reagent or product that we offer at QIAGEN is contained at one resource called GeneGlobe. And so you can find this on QIAGEN's website, and you can search for genes or pathways, and you can identify the relevant genes and you can look at pathway maps and figure out what they're related to, and access all of the species information as well. And with that, I'd like to turn the talk over to Dr. Deer, who's going to show you a specific example of using microRNA profiling to develop biomarkers.
Thank you very much, Eric. So as you said, I'm going to give, in the next 25 or so minutes, an overview of how we've used QIAGEN microscript platform to discover microRNA biomarkers for drug-induced liver injury. Now, drug-induced liver injury is a significant problem both in clinical practice and for the pharmaceutical companies. When they discover that a drug is toxic, it's often in the late stages of development, and that can lead to the drug being abandoned and a large financial loss for the company. So both from the point of view of patient care and drug development, there's a need for new biomarkers that can accurately predict drug-induced liver injury. Now, the particular group we've looked at is acetaminophen-induced liver injury. That's because acetaminophen poisoning is a big clinical problem. So the slide you have now shows data from England in 2010. And the point it makes is that acetaminophen overdose is very common. There we have data showing there are about 40,000 admissions in England. Now, numbers have actually gone up across the whole United Kingdom. So England, Scotland, Wales, and Northern Ireland, we now have around 50,000, 60,000 hospital admissions. That's a bigger number than we have for acute myocardial infarction, and a similar number, as you see, for fractures of the hip, chronic obstructive pulmonary disease. So it's a big clinical problem. So just to refresh you about how acetaminophen, or paracetamol, depending on which side of the Atlantic you are, how acetaminophen causes liver injury. So when you take a safe dose of acetaminophen, or APAP, when you take a normal dose, it's conjugated and safely excreted. However, even in a normal therapeutic dose, a small amount is metabolized by cytochrome P450 enzymes, and that forms a metabolite called NAPQ, which is potentially toxic to the liver. However, in health, this NAPQ metabolite conjugates, or it binds with glutathione and is safely removed. In overdose, however, the conjugation pathways become saturated, and more of the acetaminophen is metabolized by the cytochrome P450 system to NAPQ. Glutathione within the liver cell is exhausted, and therefore NAPQ, a toxic metabolite, binds to structural proteins in the cell, which leads to oxidative stress, mitochondrial injury, and potentially and often liver cell necrosis. In extreme cases, these necrotic liver cells will generate an inflammatory response, which can go on to cause multi-organ failure and patient death. Of note, the antidote we give, acetylcysteine, replenishes cellular glutathione, so increases the safe detoxification of NAPQ. Now, paracetamol or acetaminophen overdose is very common. The challenge is to decide who needs treatment and who doesn't. One of the big challenges is that patients come to hospital very soon after the overdose, at a time when current biomarkers of liver injury, such as alanine transaminase or ALT, at a time when they cannot exclude injury. Therefore, we're left with inadequate biomarkers. The most common one we use is simply the concentration of the drug in the blood acetaminophen concentration. So on the slide in front of you, you see a figure of the time from when the patient took an overdose to when they came to hospital. And what you see is the patients come to hospital very soon after overdose. This is within 1 hour, 2 hours, most patients are coming. And at this time point, we cannot determine who's got injury and who hasn't. So what we do in clinical practice now, both in the UK and in the USA, and in fact across the world, is we measure the blood acetaminophen concentration, and we plot it against the time from the overdose. And if you're over this line, or if you're over this line, then you get treatment. And you get the same treatment no matter how far you are over the line. Equally, if you're just under the line, you're sent home with no treatment. And this binary decision where you just get treatment or no treatment based on a drug level, it just can't be right. It can't be optimal. So what we're looking for is a biomarker. And as Eric describes, a biomarker is a biological characteristic that we can measure. And something we can measure in the circulation that tells us whether there's liver injury present or not. This will allow us to stratify treatment and target those in need. And I think the pregnancy test is the perfect biomarker. A positive result has a significant impact. It certainly does. Having a new baby, I can tell you that for sure. It adds real value. You're pregnant. It means something. It's very sensitive and specific. If you have a positive pregnancy test, you say you are pregnant. You believe it. It detects a protein that's not normally expressed in a non-invasive tissue, i.e. you have a drop of urine and it'll detect the biomarker. You don't need to do a tissue biopsy. Because the protein goes very high in the urine, there's no issue with normalization. The test is very simple, it's widely available, and it's a quick turnaround. So this is what we're looking for with paracetamol. And acetaminophen or paracetamol, I keep switching between the two, is a very good marker for discovering new biomarkers of liver injury in humans. I say this because there's a dose-dependent sterile cell death. It's one of the few models I can think of in humans where people take a drug and we know that depending on how high the drug is in the blood, there's an increasing likelihood you're going to get a toxic effect. The patients are young, which means they often have no comorbidity. Comorbidity can make biomarker studies much harder. Studies of sepsis can be very difficult if you've also got patients with diabetes, patients with existing liver disease, whereas most of the patients who take these overdoses are young with no other health problems. They have a clearly defined insult, and they have a blood measurement of the drug, so you know they've actually taken it. There's loads of patients, and from a research point of view, they've been very much neglected as a model, but they offer a real model for biomarker discovery. So just to slightly labour the point before we get on to the experimental data, I want to sort of draw the analogy with myocardial infarction, with heart attacks. So 20 years ago, when I first started as a doctor, you had a patient who had chest pain. They were risk stratified by an ECG or an EKG. And then we waited for a period of time, a day, 2 days, to detect what were then called cardiac enzymes, which were creatinine kinase, AST, and LDH. And along came troponin, troponin being a protein that's expressed almost exclusively in heart muscle. And now we can detect heart injury much nearer to the time of the pain starting. And that's what we need with acetaminophen. Patient takes an overdose, as it is now, we measure the blood concentration, and then we have to wait until liver enzymes are elevated. What we need is a new marker that allows us to rule in or rule out liver injury soon after the overdose. So what we're going to talk about today, and Eric's really described this very well, is microRNA. So microRNA are short, non-protein coding RNA species which are transcribed in the nucleus, exported into the cell cytoplasm. They're chopped up, if you like, by the enzyme dicer to form these short, around 22 base pair strands that then are loaded into proteins, such as Argonaut proteins, and the microRNA can inhibit mRNA translation, either by blocking the translation or actually by degradation of mRNA. So that's what microRNA do inside the cell. and that's been a subject of great amount of research, and it still is. But what I want to focus on are microRNA outside the cell, and particularly microRNA in the blood. So let's say this top panel is an hepatocyte. MicroRNAs are constantly released from cells. And as Eric described, they're constantly released by two main pathways. They're constantly released either in vesicles, in extracellular vesicles. And the nomenclature for these, I think, is a little bit confusing. But essentially, I think they can be broken down into microparticles, which I think of as being derived directly from the cell membrane, or exosomes, which are derived from intracellular pathways loading up and releasing these vesicles. The final group are apoptotic bodies, which are released by apoptotic cells. So together these are called extracellular vesicles, and they're either cell membrane derived, exosomes, or apoptotic bodies, broadly speaking. However, I think when you look in the literature, there's a large number of different terms used, so it's a sort of confusing field, shall we say. The alternative is that microRNAs are released from cells bound to proteins. And largely, this is the Argonaute protein that I mentioned in the slide before. So these are the proteins that make up the RISC complex that microRNAs are loaded onto to, inside the cell, inhibit mRNA. So they can be released from the cell bound to these Argonaute proteins, or they're released from the cell bound to lipoproteins. So the binding to the protein, or the encapsulation in the vesicle, protects the microRNA from breakdown by enzymes in the circulation. So when we look in the blood vessel, there are essentially microRNAs bound to protein, or they're microRNAs in vesicles. Now, circulating microRNAs, because they're very stable and because some of them are organ-specific, are very attractive as biomarkers for a wide range of diseases. The paper I have up here on the slide now, it was the first description of microRNAs as biomarkers of drug-induced liver injury in mice. So in this study, acetaminophen was used as the liver insult because it's a very well-characterized, dose-dependent liver toxicant. And what the authors found was microRNAs change in the plasma. The microRNA that changes the largest amount, the largest fold change in the plasma after exposure to acetaminophen was a microRNA called miR-122. At the same time, the authors looked at the microRNA levels in the liver, and they found that they go down. This is consistent with miR-122 leaving the liver and going into the plasma when the cell is injured. And the key thing, or certainly one of the key things this paper shows, is that the miR-122 in the plasma is more sensitive than current markers. So at lower doses of acetaminophen and earlier or sooner after acetaminophen exposure, miR-122 is elevated compared to ALT. So we've done a lot of work on this in humans over the past few years. Some of the papers I've shamelessly advertised myself on the next slide. But essentially, we've taken this preclinical work and took it into patients and asked the question, can microRNA aid us in diagnosing and in treating drug toxicity? So one of the studies I just want to go on to before we describe the profiling study is the first study we did that showed that miR-122 is elevated in humans. So we had 5 patient groups. We had patients with acetaminophen-induced acute liver injury. We had patients who took an overdose of acetaminophen but didn't get any liver injury. We had patients who had acute liver injury for a cause that wasn't acetaminophen. These were a mixed bag of causes: viral hepatitis, autoimmune hepatitis, ischemia. We had healthy controls, and we had chronic kidney disease. The microRNAs we looked at were miR-122, which was the microRNA that's expressed in the liver, that was the highest fold change in the mouse. We also looked at 192, which was another liver-enriched microRNA, and 2 controls, a heart microRNA and a brain microRNA. And what we found in a nutshell was that the microRNA 122 was highly upregulated in the plasma of patients with liver injury. So just to orientate you, this is looking at ALT, the current marker, and the group in red are patients with APAP-induced liver injury. And what we see is that ALT and miR-122 are elevated in these patients compared to the control groups of healthy paracetamol with no liver injury and kidney disease. It's important to note as well that other causes of acute liver injury also elevate miR-122. We also found that miR-192 is elevated, but not as to the same amount, the same concentration as miR-122, whereas the heart-specific and the brain-specific microRNAs weren't changed. Now, one of the questions is, is miR-122 really coming from the liver in these patients? So I'm just going to show you a couple of in situ hybridization slides. So these are patients who took a severe acetaminophen overdose and then developed severe acute liver failure and required a transplant. And this is looking at their explanted liver. And what we have here is in situ hybridization for miR-122. So to be orientated, the darker blue is miR-122, and all of this should be dark blue. But what we see is loss of miR-122 staining in the areas of necrosis. This is classic central lobular necrosis. It's not that there aren't cells there, because if we stain for U6, a small nuclear RNA, you see in these areas there still are cells, they've just lost all the miR-122. Here's a second patient, again, paracetamol overdose, severe liver failure, went to transplant, and again we see almost complete loss of miR-122, only very sort of faint staining, whereas around the areas of central lobular necrosis it's nearly all gone. So miR-122 is high abundance in the liver. When the cell's injured, it's being released into the plasma. So what we went on to do, using the microscript platform, was to ask the question, we know miR-122 is elevated. What other microRNAs are changing with acute liver injury in humans? And can we use those microRNAs to improve our diagnostic ability? So to start with the patients we looked at, we had a training set and a test set. There were two groups of patients. Patients who took acetaminophen overdose and developed liver injury, they are APAP tox, and patients who took acetaminophen overdose and didn't develop liver injury. So in the training set, there are 27 of each. Their age and sex matched. The patients with toxicity, by definition, had an elevated ALT, the current liver biomarker, and they had a degree of coagulopathy as measured by INR. So INR is a measure of blood coagulation, and it's a readout of liver failure. The test set, similarly, we had patients with toxicity and patients without toxicity, and actually in the test set, the patients with toxicity were slightly more unwell, as evidenced by the higher INR. But essentially, you've got liver injury, no liver injury, in two groups of patients, both of whom were exposed to excess acetaminophen. And the experimental plan, the experimental protocol was as follows. We started with miR-base 18, so we assayed 1,809 different microRNAs. We started by pooling samples, so we had two pools of samples. We had patients who had acetaminophen-induced toxicity and patients who didn't have toxicity, pooled together, and the first question for phase 1 was which microRNAs are present in these samples. The expression revealed 350 microRNAs that were expressed either in toxicity or with no toxicity or both. Expression being defined as a CT value less than 35. Those 359 microRNAs were then measured in each individual patient's blood plasma. So 27 patients with toxicity, 27 without, measuring 359 different microRNAs, of which we found 281 was significantly differentially expressed. So you can already say a large number of microRNAs change with liver injury. As we went on, we then developed a model to test. So here's a volcano plot of the data. So just to get orientated, the x-axis shows the fold change. So to the right, the microRNAs are increased. To the left, they're decreased. And on the y-axis, you've got the significance. The higher up the y-axis, the more significant the change. And what we see is that a large number of microRNAs have gone up. 75 were threefold or more increased with acetaminophen-induced acute liver injury. Interestingly, and I'm not sure I still fully understand this, a number of microRNAs go down in the circulation. If we look at what these microRNAs are, or what their identities are, what we see is, as we found before, as we measured before, and as per the mouse data, miR-122 was the largest fold change, the largest fold increase in microRNA. However, there were other microRNAs that were increased pretty much as much, and a number of these have not been described before. So here we've got a large number of potential biomarkers. If we look at the microRNAs that go down, we have a whole new panel that decrease with injury. Now, just to sort of as an aside, we've talked, and Eric's talked specifically about exosomes, and exosome research is definitely huge. So here's a figure taken from this paper in the Journal of Extracellular Vesicles, just looking at the number of publications that are about exosomes. And what we see with the green line is a huge explosion in interest in exosomes. Similarly with microvesicles, similarly with extracellular vesicles. And a large amount of that explosion has been driven by the recognition that exosomes and extracellular vesicles can signal between cells. A word of caution, however, and that is that there are certain feelings in the literature that microRNAs, at least in the plasma, aren't expressed abundantly in exosomes. So here we have a figure from a recent PNAS paper demonstrating that when you measure microRNA absolute copy number in the plasma, and you measure, but you count the number of exosomes using a technique called nanoparticle tracking analysis, some of the most abundant microRNAs, such as 126, have got far less than one copy per exosome. So for microRNA 126, you would need to collect about 1,000, if not more, perhaps 10,000 exosomes to find one copy. So this is just making the point that actually a lot of the microRNA in the circulation may not be in exosomes, but may be in the so-called protein fraction. And then indeed, that's what we found with the data I just presented. So if we look at our top two largest fold increased microRNAs, what we see interestingly is 122 and 885 are very tightly correlated across people. If we look at what fraction they're in, we find that this increase is in the fraction bound to Argonaut. So here we're pulling out with an antibody the Argonaut protein and measuring the miR-122, and we see that it goes up. We're measuring the 885, we see that it goes up. Interestingly, our next hit, 151, doesn't go up. It may be that that microRNA is in exosomes. If we look at tissue, this is from two patients who went to transplant, we see that 122 and 885 have very similar patterns. They're again lost in the areas of central lobular necrosis. So what this is suggesting is that 122 and 885 are both released from necrotic hepatocytes bound to this protein Argonaut. So to sort of give this data a bit of human flavour, for want of a better word, I'm just going to sort of show the profile of different microRNAs in a man who took an overdose. So this is a 49-year-old man who took a large overdose of acetaminophen, had a acetaminophen level at 4 hours of 405. I don't expect people to really remember this, but that's a high level. However, when he first came in, his liver function tests, as we use now, the standard ones, were normal. He received acetylcysteine, but he developed significant liver injury, significant kidney injury. And what we see, if we look at our microRNAs over time, we see miR-122 going up and actually coming down quite quickly. We see 885 going up, panel B, but actually staying up longer. Interestingly, if we look at some of the microRNAs that go down, we see again in this individual patient that they fall quite substantially. And this is just showing his ALT and his INR and his creatinine. So we can see over time that microRNAs are reporting liver injury, but with different profiles. But I think the interesting question for this work is, okay, but can they do something that we can't do now with ALT? So in this final bit of the study, we recruited patients from a hospital in London. I should say the data so far has been from a hospital in Edinburgh. So we've deliberately gone to a different hospital and recruited different patients. And what we've done is we've taken the blood sample when the person first came into the emergency department. And we've said, if we measure microRNA as soon as someone comes into hospital, can we predict whether they're going to get injury or not? So we had 67 patients, pretty typical age. And of them, around 13 went on to get liver injury. And what we've got over on the right side are ROC curves. So receiver-operated characteristic curves, which basically are a plot of sensitivity against specificity. And the area under the ROC curve gives an idea of the accuracy of the biomarker for predicting outcome. And what essentially we see is, if we look at ALT, which is here, and it's the third one down on the table, ALT has got an area under the curve of 0.8. So the current marker correctly identifies injury 8 out of 10 times. It's a rough rule of thumb. However, if we look at microRNA 122, and particularly when it's in combination with another microRNA, we see that it's nearly perfect, substantially better than ALT. So this is at first presentation to hospital, our microRNA signatures can tell you who's going to get injury and who's not. And this has got the potential to cause a big change to the way this disease is managed. It allows us to stratify patients and target treatment, and it allows us to send home well patients and not miss patients. So to finish off, I'll just give you an example, a real-world example. So we had a 25-year-old man. This is in Edinburgh. He took an overdose of a large amount of acetaminophen at 2:30 in the morning. He put a message on Facebook, as is the modern way, and he attended hospital 4.5 hours after the overdose. He told us the time he took the overdose, and this timing was supported by the Facebook message. So we believed the time. He had no risk factors for liver injury, and his blood acetaminophen level was below the nomogram, the line I showed earlier. His liver function tests at the time of presentation were normal, and he was discharged. That's quite normal. It wasn't a mistake. It was doing it by the book, and he was discharged. He came back to our hospital 43 hours later because he was unwell. His abdomen hurt and he was vomiting. So at 43 hours after the overdose, about 39 hours after the first presentation, his acetaminophen level had fallen, which is quite normal. It suggests he hadn't taken another overdose. However, his liver function tests were dramatically abnormal now. He had an ALT of 11,000, very marked liver injury, with synthetic function was impaired, INR of 2.1. What we did was we got his blood sample from the first presentation, and we measured the microRNA and said, could we have detected the injury? Could we have avoided him being sent home? And the answer is yes, very clearly. The microRNA 122 was around 50-fold above the upper limit of normal at 4.5 hours after the overdose. I.e., if we use this new microRNA approach, we can identify injury that's being missed at present. So I'd like to finish there. There's a large number of people to thank, not least my colleagues in QIAGEN for this collaboration. And I think at this point, we'll probably start taking questions. Thank you very much.
Well, thank you very much, Dr. Deer and Dr. Lader, for that insightful presentation. And now I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of this webinar. I have already received some questions, and so I'll start with those. This question is for Dr. Lader. Have you compared these PCR-based microRNA profiling results to sequencing-based microRNA profiling on the same starting RNAs?
Thank you for unmuting me. No, our experience to date in comparing data that we've gotten from NGS and the data that we've gotten from PCR is that they're actually fairly similar to each other. We are using some NGS technologies that are a little more advanced than what's commonly available today. So it's hard to actually relate them to each other. The other thing, though, is if you're looking to develop this as a biomarker, I imagine you want to do it quickly. And the methodologies that you need to do library construction and then sequencing deep enough to read everything will take a couple of days longer than if you just did it by PCR. I think PCR is it's much easier future for high throughput and standardization and automation than sequencing does at this point.
Great, thank you. Our next question for yourself as well. Is the LNC RNA regarded the same as ncRNA in this presentation?
Well, no. Non-coding RNA includes what are defined as long non-coding RNAs, which are above 200 bases, link RNAs, which are long non-coding RNAs that are intergenic, that are above 200 bases, Peewee RNAs, which are, I believe, germ cell specific, and they're short RNAs, but they're not microRNAs, microRNAs, endogenous siRNAs, and anything that doesn't code for a transcript. So small nucleolar RNAs are long non-coding, are non-coding RNAs as well, for example, but we didn't even talk about those today. So non-coding is one definition. Long just means over 200 bases.
Great, thank you, Dr. Lader. Our next question is for Dr. Deer. What are the roadblocks to using RNA biomarkers in clinical practice?
Yeah, I think that's a great question. I think the first roadblock is the sample preparation and the techniques we have for actually measuring the microRNA. So in the data that I presented, miR-122, I think, probably is the most developed microRNA, or it's certainly one of the most developed microRNAs. And it's not far off being ready to be used, at least in clinical trials, to stratify patients. But the big roadblock we have is that we don't have a rapid assay that we can use to measure it when patients first attend hospital. So the current techniques that were described by Eric take time and take skills in the lab. So I think the first roadblock is having point of care or certainly rapid lab-based assays. I think a second roadblock is the normalization. And there's not really, and Eric may come in on this, so you may know better than me, but there's not really a sort of agreement of how circulating microRNA is normalized. Well, by normalized, what I mean is how are we going to express the abundance of the microRNA of interest. So you could just present the CT values. However, that doesn't allow for any problems with extraction. It doesn't really tell you anything useful. One way of overcoming that is to present the absolute copy number. So do a standard curve and actually tell you how many copies of your microRNA are in your sample. And I think there's a lot of merit to that. I think as biologists, we're used to working in a concentration. So I think that's useful. I think the problem with absolute copy number is that it doesn't tell you if your sample's degraded, and it doesn't tell you anything about the extraction. So you can put a spike in, so you can take a microRNA from C. elegans and look at the extraction, look at the PCR efficiency, and then normalize to your spike in. Or perhaps the best way, perhaps not, is to find a microRNA in your sample that doesn't change with disease, that's stable across people, and express your microRNA as a ratio to that, which perhaps or should overcome the problem with extraction. But I think until the field comes together and decides on how a microRNA's circulating concentration should be expressed, that's another roadblock to clinical adoption. And Eric, I mean, what are your thoughts on normalization?
I mean, it's something that people have been thinking about for 30 years. So when we've been doing screening and when we've been doing sort of whole transcriptome-like sequencing, normalizing against, for example, in NGS, total reads, normalizing in PCR against the mean of the expressed sequences in a larger screen works really well, and it's what's typically recommended in the MICE guidelines for RNA and for microRNA. The problem becomes when you're looking to do a screen with only a couple of targets, then you are required to try to develop something that can be a normalizer, and that's for any one large experiment. You can figure that out if you have a larger data set, but it's I don't think there is such a thing really. So I think that you're right. I think maybe expressing it as copies per microliter of serum might be the way to do it. It's tough. Yeah, I'm not sure what the answer to that question is.
I think the beauty of the copies per microliter is it does allow you to go across platforms and compare the readout you get. Whereas if you normalize to something else, I think that gets a bit more tricky. But I agree. I don't know what the answer is. But I think for clinical use, as a group, we need to come to some kind of decision, because at the moment, in every paper, everyone measures it in a different way.
Yeah, and traditionally, when people have used normalizing against, say, housekeeping sequences for microRNA, they've used some small non-coding RNAs that are extremely variable in serum and plasma and really represent sort of cellular debris contamination from cells that lyse in the blood prep, and those are really horrible normalizers to use in these experiments, yet you'll see them used over and over again, presented on posters and meetings.
Great, thank you so much to the both of you. Our next question is for Dr. Lader. For exploratory studies, does QIAGEN have arrays available for unbiased microRNA profiling rather than the smaller pre-selected targeted panels?
That's a good question. Yeah, so we approach that in a couple of ways. And if you listen to the way James did his preliminary experiments, it's the way that we would recommend. In that is you have your samples that can be categorized into, for example, normal and disease. We typically would make either one or two pools of normal and one or two pools of disease of, say, up to 10 samples, do an entire micronome screen to identify what's there, and then limit, since the micronome today is over 2,000 microRNAs, do that one ugly experiment, and then quickly pare yourself down to microRNAs that are more relevant for your particular application. So you can go from 2,000 to a one array plate in one experiment, and then go on to look at which microRNAs of the ones that are robustly expressed are differentially expressed. So you're going from a full micronome to a targeted panel, looking at individual samples, to looking at a much smaller panel with a lot more samples over the course of your experiments.
Well, thank you very much for those answers, Dr. Lader and Dr. Deer. We have reached the end of the Q&A session of this webinar. If you have any further questions for our speakers, please feel free to direct them via email. That contact info is showing on your screen right now.
Thank you, everybody.
Thank you very much.
Now please join me in, oh, go ahead.
Oh, go ahead.
Now please join me in thanking our featured speakers for today, Dr. Eric Lader, Senior Director of Product Development, QIAGEN, Dr. James Deer, Senior Clinical Lecturer in Clinical Pharmacology at the Royal Infirmary of Edinburgh, Edinburgh University.
Thanks. Thank you very much for moderating.
Yep, pleasure. Thank you, Michelle.
Thank you. It's a pleasure having you. And finally, thank you to everyone for participating in today's conference. You will be receiving a follow-up email with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our future webinars. We hope you found this conference informative. Have a great day, everyone.